So this will be the last session, and I wanted to also take the opportunity to advertise our sponsor, JSTAT. I guess you know JSTAT, but yeah, it's a very important journal for our community. I am an editor, so I might be in a conflict of interest, but I think it's an important journal for our community, and it's owned by the community somehow, so yeah. You are all welcome to submit your nice papers to these journals, and I think we are all committed to do the best to provide a good editorial service to the community. This said, let's go back to what we were discussing yesterday. So I would like to conclude this series of lectures by giving you an application to proteins. And what I was saying yesterday is that the goal in protein science, let's say, is to understand, given a sequence of a protein, what is the function that the protein performs. And structure is an intermediate step that is also very important. So for going from sequence to structure, we have now AlphaFold and similar machines. But going from sequence to function or from structure to function is complicated because function is not a very well-defined concept. It depends a lot on the environment, on the context, and so on. And also because we have few data. Okay, so I made yesterday an overview of the kind of data we have. I will not repeat everything, but just very quickly. We have data from natural evolution, from which we can obtain information at large scale on the space of possible sequences that are associated to a given function. We have very local scale information coming from deep mutational scans, where you take one sequence and you do all the possible single mutations. We have experiments where people do evolution in vitro, so they start from one sequence and they let it evolve and diversify. And these are typically confined to short time scales, so they can do 20, maybe 100 generations, hopefully in the future maybe a bit more. And we have experiments where you can take two sequences that are two natural sequences, for example, or maybe one sequence and one ancestor that you reconstruct in some way. And then you can study paths of single mutations that go from one sequence to the other. Or if the two sequences are not too far away, you can study all the combinatorial space of intermediates. So in this experiment, they have two proteins that differ by 13 mutations, and so you can do all the 2 to the 13 intermediates. In this experiment, they could only reconstruct one path, but they studied the evolution along that path. And there are many data of this type that are appearing now, so that you would like to study. Okay, so now what can we get from this data? The first example I want to show is how you can infer some information on the structure from the sequence. Now, today there is AlphaFold that is doing that, but what is the idea behind AlphaFold? This is something that comes from previous studies, and in particular, so the idea is, okay, I have an alignment of sequences that are all doing the same thing, and so they have also, in particular, they have the same structure. So this is an example. This is the, okay, so these proteins are organized in families. Families are typically constructed by aligning sequences with standard alignment techniques that I'm not going to discuss, but so you can find them. And so this is an example. This is this domain, this is protein called trypsin inhibitor, and it's something that you find in many different species, from insects to mammals to bacteria and so on. So you have all these proteins coming from different species, and you can align them. And this is what you see. This is an example. It's a 53 amino acid long alignment, you see that you have gaps, but you see that you have some specific patterns that you can recognize that are also used for aligning these sequences. For example, you see that this one is very often a C, almost always a C stain. And also the second one is very often a C, okay? So you have, for example, in this family, you have these two specific C, and you have other patterns that you can exploit to align the sequences. So this is typically called conservation, which means on a certain site along the sequence, there is very often one amino acid or two amino acids or a few amino acids that are present. So to study conservation, what you can do is to compute the frequencies of amino acids at each site. A simple way to do so is shown here, and it's what we will use in the code. I will show you a code that does this. So you can do what is called the one-hot encoding of the sequences. So you represent your symbols as strings of bits. And as you see here, I'm sorry that maybe people in the back cannot really see very well, but the screen is not very big, but okay, I can describe what is there. So for example, the first sequence has a gap at the beginning. So we will encode the gap by 1, 0, 0, 0, 0, 0. Okay, so this will be. And then if you have an A, for example, the third sequence starts with an A, then it will be 0, 1, 0, 0, 0. So if you put a 1 in the first position, it will be a gap. If you put a 1 in the second position, it will be an A. If you put a 1 in the third position, like here, it will be a C. There is no B. B is not an amino acid, and so on and so forth. And so you have 21, in principle, strings that have all zeros and a 1 in the position that corresponds to the letter. In reality here, because of what I'm going to show you next, we are going to use 20 bits instead of 21. Why? Because, as I told you yesterday, there is a redundancy, of course, because there must be one of the 21 symbols. So if the first 20 are not there, then the last one must be there. So you can use 20 zeros and no 1 to encode the last symbol, okay? And if you don't do that, in your data matrix, if you use 21 symbols, then the last column is going to be 1 minus the sum of the first 20. And so you have a linear dependency between the columns of your matrix that will give you problems when you will invert the matrix, because we are going to invert the correlation matrix to get information, okay? So I'll come back to that. But anyways, so we do a one-hot encoding, but with the specification that we only use 20 bits for the one-hot encoding, and the 21 is basically dropped. And if you have all zeros, then it means that there would be a 1 in the 21 position. So you have 53 amino acids, so you will get 53 times 20 bits, and these will be the lines of your data in one-hot encoding. So if you want to count the frequency of, for example, a C in the second position, what you have to do is to count the number of ones that you have in the third column of the second position. This is the first position, second position, third position. So you count how many ones you have, and you divide by the number of lines, and you get the frequency of a C, which is basically, here you see it's basically 1, okay? So what you have to do is the operation that is done here. You just sum the columns of the matrix, you divide by the number of sequences, and you get a vector of probabilities that gives you the conservation profile. So this is, well, this is what I called actually FIA in my lecture yesterday. Here it's called PIA, but it's the frequency that you get from data. Okay, so this will tell you information on which amino acids are present on which sites. But you also have another kind of information, which is coevolution. So you can have pairs of sites that are correlated. So for example, I don't know, if there is a P here, maybe it's more frequent that there is a Y here, or something like this. So what you can do, you can count you can compute the joint probability of having amino acid A on site I and amino acid B on site J. And this is what we call PIAJB. So what you have to do is to count how many times you get the one here and the one here, and you average over the lines, and you get this. And what we are interested in is the correlation. So the correlation is going to be the difference between PIAJB minus the product of the P, because if the columns were independent, then the joint probability would be the product of probabilities and the correlation would be 0. So if the correlation is non-zero, it means they are not independent. Okay. Now, from this kind of, so this is the basic information that you can get. Then, of course, from this data you can get other quantities, other statistics, but we will stick to these two for the moment.
Excuse me.
Yes?
In the first step, how did you group the proteins? You pick the proteins, for example, in the different regions that have a similar function.
So yeah, this is the problem of aligning the sequences to construct these families, which I didn't discuss in this lecture. So the idea is that you start from a seed. So you start from a few sequences that you know to be trypsin inhibitors, for example. And this you can know because you did some experiment or because you look at the sequence and you are an expert biologist and you recognize some pattern. So this is done, this is an art of making the seed. Okay, so you start from a seed. You can do this also by aligning pairwise. You take pairs of sequences and you align them. So at some point, by some process that depends on the protein, you have, I don't know, 100 sequences or 50 sequences or even 10 you have a few sequences that you can align. Then you learn a model from this seed, and the model is typically what is called a hidden Markov model, which is a model that basically uses essentially the conservation. So it's a model that looks at the conservation profile of the sites, but it also allows to insert gaps or to insert amino acids. So it's a model where you scan your sequence and you look for conservation patterns, allowing for a bit of flexibility because you know that some amino acids can be removed or inserted by evolution. So basically, from the seed, you learn this model, and then once you have the model, you scan all the proteins that have been sequenced by humanity, and you look for hits. What are called hits are strings, sequences that the model says these are good, they have similar properties to the seed. Then the model will give you a score and you put a threshold and you say if the score is above my threshold, I say okay, this is a hit, and I put it in my alignment. And then you get your alignment. This is how it is done. And it's non-trivial. I mean, there is a whole domain of bioinformatics that is devoted to how to do this in a proper way, and there is research also on this. But this is another, this would be another series of lectures, so I'm not. So let's say this is given. Basically, today, if you go to Interpro, which is a database, you have these families and someone did the job and you can download this already. This is already done. So we consider this as a given piece of information.
Another question.
Sure.
You said that we have 20 different amino acids.
Yes.
And we can have also, we can not have amino acids. So it's, in total it will 21 different codes.
Yes.
But you said we just need 20 because the last one would be dependent to.
Yes, when I do the one-hot encoding. When I do the one-hot encoding, for example, if you have two symbols and you want to do a one-hot encoding, it's enough to have one bit because you put 1 if it is the first, and 0, it's not the first, then it's the second. So if you have 4 symbols, you put 3 bits, and you say 1 for the first symbol. You can say, okay, 1, 0, 0 is the first symbol, 0, 1, 0 is the second, and this is the third. And the fourth one, if I have 0, 0, 0, then it means there is a 1. The reason, you could do this would be the full one-hot encoding, but then this line, this column, is a linear combination of these columns, because this column here is 1 minus the sum of the first 3 columns. Right? Because, well, yes, it's 1 minus the sum of the first 3 columns. Because if there is a 1, it's 0, and if there is no 1, then it's 1. So this gives you a matrix of data that has some linear dependency between the columns, and so it's typically not invertible. And since we are going to invert the matrix, this is going to create problems. Okay? Okay, so what was the first idea that people had? This kind of dates back to the 90s. The idea was, okay, we can look, once we have these probabilities, PIA and PIAJB, we can look at correlations between sites. And the idea is that maybe if 2 sites are strongly co-evolving, so if they have strong correlations between the amino acid on one side and the amino acid on the other side, this could be a hint that this pair of sites is in contact. So if you have your protein that is, at the beginning, the protein is a linear chain of amino acids, so you will have all the amino acids that are organized like this, then the protein is going to fold. So maybe it will fold, and maybe these 2 sites, I don't know, this one and this one will be in contact. And then, okay, my drawing is not very good, but whatever, 1, 2, 3, 4. So this one and this one become a contact. And so if there is a contact, it means that these amino acids are somehow interacting in some way. And so if you change one of the 2 amino acids, it's possible that you will have to change also the other one to keep the interaction strong enough to maintain the contact. Okay? So the idea was, okay, let's look at these patterns of correlation, and let's try to see if this gives information on the contacts. So, okay, so what you want to do is to fix the I and J indices that are the ones that give you the site, but then you want to average over the amino acids that you can have on that pair. So a way of doing that is using mutual information that we defined at the beginning. So this is, in this case, the mutual information between site I and site J. So you fix I and J, and then you sum over all possible pairs of amino acids that you can put on these 2 sites, and you compare the logarithm of PIAJB with the product of the probabilities. If the sites are independent, this quantity is 0, and otherwise it is a positive quantity that will be, the more you have correlation, the bigger it will be the mutual information. Okay? So now what you do is you take all the pairs of sites. For each pair, you have one number, which is the mutual information of that pair, and then you sort the pairs from the one that has the biggest mutual information to the smallest. So what is shown here, in this case, for this protein, for the trypsin inhibitor, the structure is known. So this is the structure, it is shown here. And what is shown in this plot is the following thing. So you take your pairs. So for each pair, for example, 1, 2 will have some mutual information. So there will be M1, 2, then you have 1, 3, and you have 1, M1, 3, blah, blah, blah. And then you will have 52, 53 that have some M. Now you rank according to M. So you take the pair that has the biggest M, the second biggest, and so on. And you take the first K pairs, the one that has the top mutual information. So the x-axis here is the number of pairs, okay, that have the biggest M. So for example, when it is 10 here, you are taking the 10 biggest mutual informations, pairs. And what is in the y-axis is the fraction of those pairs that correspond to contacts, okay? What is a contact? There is a convention, because what is exactly a contact is It's not well defined, but typically one says that two amino acids are in contact if the distance is smaller than 8 angstrom. The distance between the carbon atoms in the center. Anyways, what is it? I mean, if they are close enough, you say that they are a contact. So from the structure, you can get the structure from experiments, and from the structure you can get the position of the atoms, and then you can decide that some sites are in contact. So among the top 10 mutual information pairs, all of them are contacts. Then you increase the number of pairs, so you take the top 20 here, this is log scale, so you take the top 20, and you find that 80% are contacts. Then you take the top 100, and you find that 50% are contacts, and you can keep going. So you see at the beginning, all the top mutual information pairs are indeed contacts. This is what you expect, and so this is an indication that the hypothesis we made is correct. So if two sites are in contact, they are likely to be strongly correlated. I mean, the amino acids that you put on those sites are likely to be strongly correlated. So the two curves here are, the full line is, I mean, the dashed line is all contacts. The full line is you take only contacts that are a distance bigger than 4 along the chain. Why? Because of course, I mean, amino acids that are close, that are neighbors on the chain are in contact. So it's a bit trivial, right, that these are in contact. So when you try to predict contacts, you want to predict non-trivial contacts that are contacts between sites that are distant along the chain. So you typically, these are also again a bit arbitrary, but you typically restrict to things that are bigger than, that are a distance bigger than 4 along the chain. You see, it doesn't change much in this case, but okay. Now, here it's the same thing, but with a correction, that in this case it's not really useful, but that sometimes is used. It's called average product correction, where you construct a score that is given by the mutual information minus, here this notation means mi dot means that you sum over j mij, and here you sum over I mij, and here you sum over both. And the idea of this score is, of this correction, is that basically you know that in 3 dimensions one sphere can have 13 neighbors, I think, and so you cannot have more than a certain number of contacts just by geometry. So basically, if a site I has too many candidate partners, so if a site I has strong mij with many j, there is something wrong, so you want to lower the score. So this is a way you can think a little bit, and this is a way to remove, to downgrade a little bit the sites that have a lot of candidate connections, okay, just to take into account of this constraint. But it's an empirical recipe that is, well, motivated in this way, but okay. Anyways, in this particular example, it doesn't change much, so you can see that the two curves are very similar. What you see here, to get an idea, a more visual idea, is the structure, and here you have the top 30 mutual information pairs. So you stay here at 30. When you are at 30, you know that 60% of the top are contacts. So here the color code is the non-intuitive one, so red is good, it's a contact, and green is not a contact. So you see that among the top 30, there are some contacts, but there are also a lot of false positives. So can you do better than that? Yes. How do you do better? By doing what we discussed in the previous lectures. You should take into account the fact that correlation is not causation. So two sites can be strong. There could be, I don't know, maybe this site can be in contact with some other site here. And so this one and this one might be correlated, but just because they are both in contact with the third one. So you want to go from correlations to couplings. So this is where inferring a graphical model is useful. And so we have seen that one way to do that is to assume that the variables are Gaussian and then invert the matrix, or assuming that the variables are categorical, which is correct in this case, and then do mean field. And in both cases, what you get is that the matrix of couplings, so the graphical model, is given by the inverse of the matrix of correlations. So in this specific case, what you have to do, the correlation matrix is a matrix that is of size 53 times 20 times 53 times 20. So you consider your one-hot encoding here as one data point. So it's 53 times 20. And so this is considered as a single index, right? And then you do, so then if you call YIA M, sorry, YIAM is going to be, M is the index of the row and YA is the index of the columns. Then this defines this matrix Y. This is a matrix of size M times times 53 times 20, where this is the size, this is L, the length of our strings of bits. And then the correlation matrix is just 1 over M Y transpose Y, as we did for in the lectures. I mean, you have to subtract the mean first and divide by you have to standardize the Y. So you have to divide by the mean and, sorry, you have to remove the mean and divide by the variance. Okay, but the variance is just equal to the mean because these are binary 0, 1 variables. Okay, so once you do that, you can invert the matrix. So you can invert the matrix and you get J is minus C2 minus 1. And so J is a matrix of size L by L, so 53 times 20 times 53 times 20. So it's going to be a matrix JIA JB. And again, we are not interested in the coupling for each amino acid pair, but we're interested in a coupling between pairs of sites. So in order to average over the amino acids, what you do, you define this object that's called the Frobenius norm. So you just sum the squares of the J, because the J can have positive or negative signs. So you can have, if the J is positive, it means that these two amino acids are likely to co-appear together, to co-occur together. If it's negative, they are likely to not be present together, but both signals are good, so you want to average the square.
Sorry, why do you take the minus sign there? J is equal minus C to the minus 1?
It's just a matter of convention. Here I mean, the minus can be or not be there. I mean, when I put the minus sign is that I'm considering that the model is a Boltzmann distribution and J is... so if my probability is the exponential of plus J, sigma J, and then I have sigma IA, okay. If I call sigma IA a variable that is 1 when amino acid A is present on site I, if I put a plus here, then my Hamiltonian is minus this. And this means that positive coupling means ferromagnetic interaction, so they are likely to be together. Negative coupling is... So this is my usual convention, but sometimes you can put a minus and then you have a plus. Anyways, since for this particular application, since I'm going to average the square of the couplings, it doesn't really matter. But usually we use the convention of physics. So positive coupling is ferromagnetic, you want to be together, and negative is antiferromagnetic. Okay, So now, for each pair of sites, you have another score that is not the mutual information, but the average of the square, the sum of the squares of the couplings of that pair of sites. And you can do the same thing. So you rank the pairs by the score, and you fix a number here, for example, 10. So you take the top 10 sites that have the biggest couplings, and you ask, is this a contact? And yes, it is. And you see that, okay, this is for all contacts, and this is for contacts that are far away, sites that are far away. And, well, if you just use F, you don't do much better than mutual information in this case. But if you correct with this APC, so you do Fij minus the sum over j of Fij, sum over I of Fij divided by, you see that you get a big improvement. So now here, out of the first top 40 contacts, the top 40 pairs according to this score now are all contacts, and then it decays. But you see then here the top 30 Frobenius norm pairs are all contacts. Okay, so you can have a big improvement. Yes?
Can you repeat what is on the y-axis?
Yes. So I fix a number that is the x-axis, for example, 20. I take the top, I take the 20 pairs ij that have the biggest f, and this is my 20 predictions for contacts. Then I take the structure and I look, are they contacts? So are they at distance smaller than 8 angstrom? And if yes, then the y-axis is the fraction of sites that are true contacts. So it's the fraction of true positives. Okay. So when this, up until the point where this is 1, all your predictions are correct. So here the first 40-something predictions are correct. And then, like here, if you do 100 predictions, you have 20% of false positives. Okay. Okay, so you see that you have a big improvement. There are some technicalities that we can see later in the code. You have to use a pseudo count. It's a sort of regularization. So a pseudo count means that you, okay, I will show you. And you have to, because you have this invariance of the model under this over-parametrization thing, this gauge transformation, as we call it, you have to choose, it's better to choose a gauge that minimizes this f. So you do a transformation to what is called zero-sum gauge, that is a choice of these j's such that sum over a of j I a j b is equal to 0. Okay, so you fix, I told you that you can add a constant to each line and column of the matrix. So you add the constant such that you set to 0 the average of j over a or b in such a way that your matrix is as centered as possible, and the part that you remove goes in the fields because it's not a true coevolution signal. So these are technical things. Now, before I show you the code so that you can do it yourself, I just want to mention that, okay, this was done in the 2009. The first paper, I think, is from Martin White and co-workers, Terry Hua and others. There was no AlphaFold at the time, but so it was a promising result. And AlphaFold is doing many things, but one of the first things that it does when you give to AlphaFold an input sequence, it will look for an MSA. So it will do an homology search to construct an alignment. And from the MSA, it will compute C, the matrix C, and it will invert it to get J. And then this J is fed into the rest of the layers. Okay, so AlphaFold is using this information, and indeed, when you give to AlphaFold the sequence that has no homologous ones, so if you give a sequence where there is no family, the performance is reduced. Okay, so this information is really important. This coevolution information is really important for AlphaFold to predict the structure, because basically, I mean, if you know, like in this case, if you know that many sites are in contact, then this gives you a lot of constraints on the structure, so it helps a lot. So, okay, this is the message. So the statistical properties of these natural sequences contain information about the structure, and I want to show you how you can do it yourself. So if you're interested, you can go to this notebook that you have in the folder. And so here you have, so you can input a FASTA file that is, for example, here this PF14. So let's see if it works now. Anyways, why it, so this is the kind of, yes. Okay, so this is how it looks like when you read the FASTA. So it's the file I was showing before. Here you see that you have how many sequences you, okay, I will not run all of it, but you have 8,871 sequences of length 53. So you can, okay, in the FASTA format you have a header that is this one, and it typically tells you something like where does it come from. So this is a domain, so here you see it's from 990 to 100042, so it's a domain in a longer protein. So you can drop the header. This is how your alignment will look like. You convert into 1/0, and this is what you get. And then the only thing you have to do is to compute the frequencies, and so the P is the sum, you count the ones in each column, and the PIAJB is this. Sorry, this is P, because you have to remove the mean. So then you to get C, you do just this operation. You take P, you take this, and you subtract the outer product of P. So, okay, you can think a little bit about the indices, and you will find it's correct. Now, I was saying you have to put a pseudo count. This is a regularization. So what you do is basically you have all the lines of your matrix, and what you do is to add, to say that PIA is replaced by some alpha PIA plus 1 minus alpha times a uniform distribution, 1 over Q, where Q is 21. And then you can tune alpha. Okay, so alpha is a kind of regularizer. You add a little bit of uniformity, because the idea is that maybe because you have few data, maybe some probabilities can be 0 just because you never see an amino acid, but it's because you don't have enough data, so you add a little bit of uniformity to your distribution. So it's a sort of L2 regularization where you try to avoid big parameters in your model. Here, actually, this has been found empirically that you actually need a quite big regularization. So this alpha is chosen to be 1 alpha, which is a bit strange. And this is because the mean field approximation is not a very good one. So you need a strong regularization. Actually, if you do pseudo-likelihood maximization, you don't need this big regularization, and you can get similar results, actually better results. Anyway, so this does the pseudo count. You have to do this for the P, and for the P2, for the joint, you have to do a regularization a bit more carefully, but you can look here, and you can. It's also described in the book, so I will not go into the details. And then here you have all the routines that do the mutual information calculation, and then you just invert the matrix here. You just invert, and you compute this Frobenius norm, and you can get these other files, these other figures. So if you want, you can play with this. You can look at what changes if you change, for example, the regularization. You can take other families. You can do what you want. So this is one notebook that you have in the folder here. Now, okay. So is it okay up to this point? Do you have any questions on this part? So this is a first hint that you can do something with this data. You can use this data to get information on structure. Okay. Yes.
I don't understand. If two proteins are touching and then they co-evolve, it's because
If two.
when we say that if two sides co-evolve, it's because they're touching.
Mm-hmm.
I don't get why. Because if the touching is functional, I can get it. If that doesn't happen, it stops becoming functional.
Yeah. The idea is that basically, if the two sides are touching, then it means that there is a physical or chemical interaction between the two amino acids. So if you mutate one of the two, for example, you can change the charge. So if you have a positive amino acid and you mutate into a negative one, negatively charged from positive, then on the other side, maybe you want to compensate. Because if you have two positively charged amino acids, they will repel. So if you...
But if the protein is non-functional, it's not a problem that they repel.
Exactly, but the point is that we are using, our data are natural proteins that are functional.
But we can also have natural proteins that are non-functional.
No. The idea is that the proteins that I'm putting in the alignment are all proteins that are known to be, well, not functional, but they are known to fold into this structure. So at least they fold, and then they are also functional. But now we are just looking at the structure. So all these sequences fold into this structure, the same structure. So if they fold, it means that the contact is there.
So we have to check this in vitro, like we have to check that the...
No, because we are assuming here that these are good sequences that nature already selected for us. So we don't need to check. I mean, okay, you could suspect that some of these are like false hits, but in general, in these databases, okay, in these databases you have many sequences. You can have or not a structure. In this case, you have a structure. You don't have this. Here you have 9,000 sequences. You don't have the structure experimentally for all of them, but you have a few that are measured, and they have the same structure. And the others, you assume that they have the same structure. So the assumption is all these sequences, if you could test them, they would have the same structure. So if these two sides are in contact in one of them, they are in contact in all of them. And so if there is a mutation on one of the two, you should expect a compensatory mutation. Did I get, I'm not sure I got your question right, but.
No, it was because last year we had a project on this.
Yes?
Both functional and non-functional proteins.
Ah, well, then it depends on what you want to do. Yes, you can have, okay, I know what you're talking about, but this is another problem. So, okay, this is function. Now we are doing structure.
Because last year we did the same, but they weren't functional. So
But yes, but it was a different. Okay, I will come to that later. For the moment, the only thing we are doing, we are not talking about function. We are just talking about structure, and we are assuming that all the natural sequences are working well. Are working well in the sense they are folding. They are folding in the species that uses them. So if for example, if this one comes from an insect, I'm assuming that in that insect it is folding. Now we will come to what you are saying. So yeah, that's the second point. The second point is now we we have our couplings, and so we can give a probability to a sequence, okay? So as we said yesterday, I can say, okay, the probability of a string of amino acids can be written as an exponential of sum over IA of delta, sorry, H IA delta I, sorry, delta sigma I A plus times this divided by a partition function. And I can, so if I learn from my data the couplings and the fields, then I can assign a probability to each sequence of the same length. So what can I do with this probability? I can try to check if this model knows something about whether these sequences are functional or not. And so this is generative modeling. The idea is, in generative modeling, I mean, you know it, I start from a training set of examples that I think are good. For example, I have many images of human faces. So I'd say, okay, this is what I want. So these are good images. I learn a model, typically by maximizing the likelihood of my training data. So now I have a probability distribution. And then I can generate one new sample that should be an image of a human face. How do I evaluate the quality of the generation? This is very tricky because, I mean, in images you can just look at the image and do you think this is a human face? Well, yes. But in proteins it's not totally trivial to check what you're doing. And also you would like to be, if you have a good generative model, you would like the samples that you generate to be different from the training sample, because of course a trivial way of generating new data is just to generate data that are identical to the training set, but they're not new. You generate human faces, but they're not new. So you have to check whether your data are look like human faces, but are also different enough from the ones that you have in the training set, and this is tricky. So in the case of proteins, how do we do that? I already said it. We start from our MSA, for example, the PF14, the trypsin inhibitor. We learn the model. The model has this form. We can learn the model in many ways, as we discussed yesterday. And so for the contact prediction, we can do mean field, or we can do pseudo-likelihood maximization. If you want to generate things, mean field is not good. And pseudo-likelihood maximization is not good, so you have to do Boltzmann learning. But okay, you do Boltzmann learning. And you can check that if you do Boltzmann learning, you can generate sequences that look statistically very similar to the ones in the training set. I will show you measures for this. And also that are distinct enough from the ones in the training set. We are going to use these Boltzmann machines because they are simple. So, I mean, now this was just to, typically people will say, okay, but why don't you use language models or more deep nets, or they are better? No, I mean, they are not necessarily better. I mean, they can be better for some things, but they're not always better. So in this case, they are not, at least for what I've seen up to this point, they are not really better than a simple Boltzmann machine. But the Boltzmann machine has the advantage that is more interpretable, easier to train, so you save energy, and it's very simple to implement, so you can have a code that everyone can use. So I don't see, for the moment, the need to invest much more energy or money to have different models, but maybe I'm wrong, and it will be the case in the future. So for the moment, we will stick to Boltzmann machines. And so this is, these are data taken from this paper from a few years ago from Martin White and also Rémi Monasson, Simona Coccora, Amaran Ganathan, and many other collaborators. Russ was the first author that carried on the study. These are maybe the data that you have seen last year. So the point here is different from structure. So the point is, this is another protein. This is the chorismate mutase. So the chorismate mutase is an enzyme. It's 96 amino acids long. And it is used by E. coli in a pathway that is needed to produce some amino acid. So if you grow the bacteria in a medium where this amino acid is not present, the bacteria have to produce it, and they need a functioning chorismate mutase. Otherwise, if you give to the bacteria the amino acid in the medium, they can eat it, and they don't need the chorismate mutase. So you can select your bacteria by starving them of that amino acid in such a way that they have to produce it. But now this is really function. This is not structure. So you can have a protein that is able to fold in the correct structure, but maybe for some reason it doesn't work in E. coli. So this is shown here. So what they did here as a first test of the platform. So here you have, again, you have an alignment of sequences. You have, I think in this experiment at the time they had of the order of 1,000 sequences. So here you have 1,000 sequences that come from different species. You take the DNA, I mean, you synthesize the gene, you put it in a plasmid. The plasmid is put into the bacteria. So now the bacteria have this gene instead of the original one of E. coli. They have another one that you take from another species. And you let them grow in this medium where they don't have the amino acid that is produced by the chorismate mutase, so they have to produce it. So if the chorismate mutase works, but now works is not just folding into a given structure, it means it folds, it can interact with the other partners that are needed in this pathway, and so on and so forth, then the bacteria will survive. So what you have here on the x-axis is the enrichment. So they put all the mutants together. So you have a library of here 1,000 natural chorismate mutases taken from all possible species. You put them, you make a culture with bacteria that have different genes, different chorismate mutases, and you let them grow, and you measure after some time the enrichment of each of the mutants in the population. So if the enrichment is, let's say, this is normalized to the wild type. The wild type means the E. coli with its own chorismate mutase. So if it is close to 0, it means this mutant is as good as the wild type. If it is -3 here, it means that basically that mutant disappeared from the population after some time, so it's not working, okay? If you if you make a histogram of this data, you see that it's quite bimodal, okay? So you have I mean, you see it from the scatter plot, but you see it better in a histogram. So they put a threshold here and they say, okay, below 10 to the -1, it's, okay, actually, I'm not sure if it is 10 to -1 or e to the -1, but anyway, you put the threshold here, should be 10 to the -1, I guess, but if you put a threshold here and below that threshold you say, okay, this mutant is not working. So in this sense now this is non-functional in the sense that you are taking a protein that comes from another species that is able to fold and function in that species because it was selected by nature, but once you take it and you put it in E. coli in that specific experimental setting, the E. coli dies. And you don't know really why it's not working. It could be because the machinery of E. coli is not able to express that protein, or maybe it is expressed, it folds, but then it's not binding to the other protein of E. coli that is in the pathway, or any sort of reason, okay? So there can be many reasons. Anyways, 30% roughly of the natural proteins in the alignment work in E. coli, okay? And this is the x-axis. The y-axis is the energy that the model gives to these sequences. Now, the model is trained on these sequences. So because you train the model by maximizing the likelihood, so minimizing the energy of the natural sequences, as you expect, all the natural sequences have low energy, okay? So here you have low energy, 30% of the sequences work. Now, what they did here was to sample new sequences from this probability. When you sample, you can change the temperature. You can put a temperature here in front. And in this way, as usual in statistical physics, you can change the energy, okay? So let's say I say this is my energy, so this is e to the minus energy. So now I can sample from e to the minus beta times the energy. And by changing beta, I can change the energy of the sequences I'm sampling. So in this way, they could sample sequences at different energies. And then they tested them in the same experiment. And what you see is that among the sequences that have low energy, where low, here there is a threshold that is more or less the biggest energy of the natural sequences. So among these, again, 30% are functioning. Is this trivial? It is not, because if you think to the number of sequences that you have 21 to the 96 sequences, so it's a huge space. If you take a random sequence of 96 amino acids, it will never work. So the fact that the model is able to generate random sequences such that 30% of them work is really non-trivial. And in particular, what they show in this experiment is also that if you don't put the J and you just use the conservation profile, the success rate is 0. How many sequences you can generate? This, to have an idea, what you can do is to compute the entropy of the model, okay? So let's say you fix the temperature to 1. 1 is more or less the temperature at which you sample in this region here, because it's the training temperature. And you can compute the entropy. If you compute the entropy, what you find is that this entropy is of the order of the entropy divided by the number of amino acids is about 1.5. Now, the uniform distribution has an entropy equal to logarithm of Q, which is 21. And 1.5 is about half that, is about logarithm of 21 over 2. So this means that the number of sequences that you can generate from this model is about, so the number of sequences that you can generate that have the right energy is of the order of Q to the L over 2. Because the entropy is divided by 2, so the number, which is the exponential of the entropy, is the square root. This is still huge, okay? If you put the numbers, you get 10 to the 65, okay? So you have of the order, the model can generate of the order of 10 to the 65 sequences, out of which the experiment tells you 30% are functional. So the number, this is the number of generated of the sequence, this is the number of sequences you can generate. The number of functional sequences is going to be 0.3 times 10 to the 65, which means still huge. Okay? So the point is, if the success rate is of order 1, then compared to the entropy, it's nothing. So this means that,
Sorry?
yes,
How did they test they are functional?
this is the test. So you generate 1,000, and you see how many of them are functional. And
They actually synthesize them and
yes. So you sample from the model. Now you have sequences. You synthesize them as you did for the natural ones. The natural ones, you have to, you know the sequence because someone sequenced the chorismate mutase in some other organism that is not E. coli. So what they did here is you synthesize this natural sequence and you put it in a plasmid, and the plasmid is put in E. coli, and then you run the experiment. So you do exactly the same thing here and here, except that here the sequences are the training ones that you get from nature, and here you're generating new ones. And you do it for 1,000, and because the success rate is of order 1, it converges to a value that you can estimate it from 1,000, but then you can say, okay, if I did 10,000, it should be the same. Okay. So you conclude that there is a huge space of sequences that work as well as the natural ones. Okay. This is interesting because it tells you that nature is not really, is not solving a very difficult problem. I mean, there are a lot of solutions to the problem of making a functional chorismate mutase. So if you sample, if you have an evolutionary process that is going to sample the space, it's going to converge pretty fast to some solution. But now the question is, how different are the artificial ones from the natural ones? Because, of course, one could say, okay, but if the model is just proposing 1 or 2 mutations. So this is this plot. In this plot, you have, in the x-axis, the identity to the closest natural sequence. And what you see is that you have a range. I told you that if you take 2 typical natural sequences, so if you take 2 of these sequences at random, the identity is close to 20%. They have 80% of the sites that are different, so you are more or less somewhere here. Now, when you generate from the model and you look to the closest one, you see something like this, which means that if you make a sketch of this, it means really that the energy landscape, if you make a plot of the energy as a function of the sequence, this is, of course, the sequence is a high-dimensional thing, but it's a sketch. But you really expect to have something like this, where the natural sequences are staying like in the low energy part. And when you generate artificial sequences, the more you go far away from the natural sequences, this is the energy, and the more the energy goes up. So if you move away from the natural sequences, you go up in energy, which is not surprising because the model is constructed to give low energy to the natural sequences. And the more you go up in energy, the more it's unlikely that your sequences work. So the red dots are the ones that work, and the gray dots are the ones that don't work. But you see that you can go as, I mean, that these wells are flat enough here. My drawing is not very good. They're flat. So you can go up to 40% mutations away from the closest natural. Here you have a few that are even at 60%. So you are generating things that are very different from the natural. Typically here, this is a 100 sequence long. So if you have, here you have 40 mutations, and you can go up to 60 mutations to the closest natural, and still it works. Okay? So this is something that I think you should always do when you try to make a generative model. Check the distance of what you generate from the closest training point. And people not always do it. So it's important to have an idea of how far you are from the training set. And here you can go quite far. Okay, is this clear? So in this sense, then what I think you did in your project last year was that once you have this data, you can use it to improve the model. So you can say, okay, now I tested these sequences. I know that these ones are functional and these ones are not. So I can improve my model by integrating this information. So just to give you an idea of the kind of things you can do with the, sorry, I forgot. Maybe before going to this, I wanted to show you, if you want to use this, you have this other notebook in the drive. So, okay, maybe I will not, well, let's try to run it. Let's see if we can run it. For PF14, just to give you an idea. So we have this package to do the Boltzmann learning. That is a Python package that you can use. So something went wrong. What's the issue? So I read the same data. I read the PF14 alignment. And then you can install this package. It's called ADA BMDCA Pi, where DCA is direct coupling analysis. BM is Boltzmann machine. ADA stands for adaptive, because you can also, in this package, you can also use this procedure of adding or removing couplings to make your model sparse, which we are not going to do here, but okay, so you can install your model, and then okay, it's done. I just want to run it to show you what happens when you run it, but so okay. Hopefully you can get a GPU from Google. And then, okay, you can import the data. You see here it tells you that you have 8,871 sequences of length 53 with Q equal 21. And now you prepare the system. So something that I forgot to say is that when we do this Boltzmann learning, we give a weight to the training set, and the weight is typically calculated by checking how many sequences you have around you at distance smaller than 20%, which is a convention, but it's found to work well. So you count how many sequences you have that are closer to 20% identity to you, and the weight is 1 over this number. And the idea is that you want to give less weight to sequences that are close to many other sequences, because like if you have many sequences here, you want to downweight them a bit, because the idea is that these are all maybe related by phylogeny. You can have bias in the alignment. You can have a phylogenetic bias that maybe there is a big cluster of sequences that are close, or maybe for some reason people sequenced more of this because it's interesting. I don't know, it comes from a species that is well studied, so you have more sequences. So you have this kind of biases, so you want to downweight a bit the regions that are too populated. So this is done here, and then you get an effective number of sequences that is the average weight that you have, 1 over the average weight. So you get an effective number that is, here it's about half the original one. Okay, we put a small pseudo count that is, in this case, is very small, is 1 over 4,000, compared to mean field where you put a pseudo count of order 1/2. And then with this pseudo count, you compute the FIA and you compute the empirical frequencies. Now I will call them FIA and FIA-JB. So these are the expectation over the data of delta sigma IA. So you do this once at the beginning of the learning, and then you run. So here you can choose how many Monte Carlo chains you want to use to sample from the, to compute, because you remember, in order to do the Boltzmann learning, you have to compute these quantities from the model, and to do that you need Monte Carlo. So you can choose the number of chains, you can choose how many Monte Carlo sweeps you do at each gradient update, which is an important parameter, because when you do Monte Carlo, you want to be as close as possible to a good sampling, so you want to be in equilibrium. And in order to equilibrate, you need to do a certain number of sweeps. So this number depends a lot on the family. And if you're not careful, and if this number is too small, you can have a problem of convergence of the Boltzmann learning. But okay, this would require a long discussion, and again, you can check. I can give you references if you want. But anyways, for this PF14, the Monte Carlo converges fast enough, so 10 sweeps is good. You can fix a target Pearson, which is the stopping criterion. So the learning will stop when the Cij of the data and the Cij of the model have a Pearson coefficient of, so as soon as it becomes bigger than here 0.9, you stop. Usually we do 0.95, but if I do 0.95, it will take half an hour to run, so well. And then you have to fix a learning rate that you can choose. Here is 5% is good enough. And finally, you can choose a sampler. You can use Gibbs or Metropolis here, and well, yes.
Can you repeat why we're doing Monte Carlo?
Yes, because when we do Boltzmann learning, we want to update the parameters of the model. So we start from, in this code, we typically start from a model that has 0 couplings, and the fields are fixed by the frequencies of the amino acids. And then the update equation is something like HIA t plus 1 is equal to HIA of t plus a learning rate times the expectation of this, let's say, FIA minus PIA of t, where FIA is the expectation over the data, and PIA is the expectation over the model. So you need to compute this. So you need to compute, these are essentially the magnetization of the model. So you need to compute an average from a Boltzmann distribution that is complicated because you have parameters that depend on the sites and so on and so forth. So you cannot compute it exactly, so you need to sample. So the Monte Carlo is done in order to compute these quantities. Basically, you generate, here you have 10,000 chains, so you run the Monte Carlo with 10,000 chains, you generate 10,000 sequences, and then you compute the probability that over these 10,000 sequences that you have amino acid A on site I, and this is your estimate for P, okay? Okay, now the training works like this. Okay, perfect. It's not working. Missing 2 required positional arguments, but they are there. Maybe there was a, let's see. Sometimes we update the package. Okay, there was an update of the package. Okay, so you see here how it works. The training runs with epochs. Each epoch is one of these t. So now t is 35, 36. Every epoch is an update of the parameters. At each epoch, you do the Monte Carlo. And what you have here is the log likelihood that is given by the log of this averaged over the data. And you have the Pearson here. So you want to reach 0.9, so here we are at 0.5, and it's increasing. The log likelihood is actually the true log likelihood, because in this code, we are also able to track Z, and the way we do Z is that because you change a little bit the parameters at each step, you can write, you have an expression for log Z at time t plus 1, which is log Z at time t plus a correction, and you can compute the correction. So while you learn, you can also track Z, so you can have the full likelihood and the entropy of your model as a function of time. So, okay, something that I can tell you is that if you plot, here the plot will come later, but if you plot the Pearson, if you plot 1 minus the Pearson versus time, you typically get something like this. So you want the Pearson to be as big as possible. You want it to go to 1, ideally. So at the beginning, it is 0, because you don't have any coupling, so the correlations are random. So it typically does something like, this is in log scale, and this is in log scale. It will typically do something like this. So it starts, the Pearson will start growing, and so this goes down. And then it will go to 1, so 1 minus rho will go to 0 as a power law of time. So at the beginning, it's fast, and you see, okay, it's converging very fast, but then you see that now it's saturating a bit, and it takes quite a lot of time to go down, okay? You can play with the learning rate, you can do things. We try to optimize the code as much as possible, but we didn't find a way to go faster, so you have to wait. And so, well, you see, now we are about 0.8, and in 2 or 3 more minutes, it will converge. So once you converge, okay, this is a plot. Okay, this is the plot I just made. So these are the epochs. This is for another run, but okay. And this is 1 minus the Pearson. So you see, you get this power law behavior here. We can maybe, well, this will take 2 or 3 more minutes to converge. So in the meanwhile, I can show you what you can do once you have the data. So first of all, you can check, once you learn your model, you can check that you did right by running the Monte Carlo again and computing the time it takes to decorrelate from an initial state. So this is shown here. And you should check that the number of sweeps that you used in the train. So I forgot to say, 1 Monte Carlo sweep means that you try to change each amino acid once. So if you have 100 amino acid, 1 Monte Carlo sweep is 100 Monte Carlo steps, 1 per site. So you should check that the number of sweeps that you used in the training is, this is, what is shown here is the sequence identity between the sequence at time 0 and the sequence at time t, and this is compared with the sequence identity between the sequence at time t over 2 and the sequence at time t. So if you decorrelate, then 0, t over 2, and t should be independent, and so these two quantities should be similar. You see at the beginning they are not similar, and when time increases, it goes down. Okay? So, sorry, I said it in a wrong way. So, sequence identity at time t here is the sequence identity between 2 different chains at time t. And this is the same chain at time t and t over 2. So, if you are doing your Monte Carlo, you start from, the chains typically start from either from the natural data or from some sample that you give. It depends, I mean, but say you start your chains, then your chains will evolve in time, and you look at the sequence identity between 2 different chains at time t, and you look at the sequence identity between the same chain at time t and at time t over 2. So if your time is bigger than the mixing time, this should be an equilibrium sample, this should be an equilibrium sample, and this should be an equilibrium sample, and they should be all independent. So if this, then these 2 quantities should be the same. So this is, if the time is long enough, you see that they are the same. But this is for another protein, so let's see if I can update. It's still, it will take a few, maybe 1 more minute, and then we get our result. So once we have that, you can measure a few things to check that you are generating right. So this is a PCA plot where we have in black, we have the natural sequences. So we take the natural sequences, we compute the Cij AB matrix, we do PCA, and we project on the first 2 principal components. And then we do the same with the generated sequences that are projected on the principal components of the natural sequences. So the principal components are the same here, and I'm projecting the 2 datasets. And you see that they are very similar. Also, you can make a histogram of the principal components, and you see that they are similar. And then you can compute the 1-point frequencies, and it's not shown because they are basically perfectly correlated, because the fields are always very easy to fit. So the conservation is always very easy. What is more difficult is to fit the Cij. So this is what you get typically at the end of the training. You get, this is the correlation between the Cij of your natural data, the training set, and the Cij of the generated data. And then you can compute, and this is fit by the model, right? So this should be close to the diagonal by construction. But then this is not a fit, because these are free site correlations. So you take ijk, and you ask, what is the probability that I have 3 amino acids in 3 different sites? You do it for many triplets. Here we choose, because there are too many triplets, we just choose a few at random. And you can see that you have a good fit also of the 3-point correlations that you didn't put in the model. So this is just the model is able to catch.
Sorry?
Yes?
You say that the package also can compute the 3-point correlation if you
Yes, the package is, there is a routine in the package that does it. And then here you get a histogram of the energies. I want to show this because this is important. This is the histogram of the energies of in blue you have the natural ones, and in yellow you have the ones that you generate. And you see this is not perfectly matched, because by construction the model is trained to maximize the likelihood of the natural sequences. So it always gives a slightly lower energy to the natural sequences, which means the natural ones will be always a bit on the bottom of these wells, and the ones that you generate will always be a little bit higher in energy. You can regularize this a little bit. So if you make the model sparse, this will improve. You get a better match between the two. But it's never perfect, because the model is always a bit aware of the fact that the blue are the training set. So by construction of the maximum likelihood. This is done. So we can see quickly, and then we conclude. So here it finished, so we can do this plot. Okay, this is for the PF14. This is the Pearson versus the number of epochs, and you see the power law. It's even more visible than for the other family. And now you can sample. So when you sample, you have to tell how many sweeps you want to do. Now we are fixing the model. The model has been trained, and we are generating new artificial sequences from the model. So this will run the sampling for a certain number of sweeps. And so at the end, you get your artificial sequences that you can compare with the natural ones. Now this will take a little bit of time. So while it runs, we can see the last things. So this I was saying is the histogram of the energies. And here you can check the distances between sequences. That is important to know if you are generating well. So here you have MSA1 is the training one, the natural ones, and MSA2 is the generated ones. So this is the histogram of pairwise distances. This is not the PF14, so it's longer than 53. but when we will redo it with the PF14, it will be different. But anyways, you see that if you take two training sequences at random, they will have a certain distance. And if you take two generated sequences at random, they have the same distribution of distances. But what is more important is that you can compare, you take one of the sequences you generate, and you look to the distance between this sequence and the ones in the training set, and you get a histogram like this. And then you do the same, but you take one sequence of the generated set, and you look to the closest sequence in the generated set. And if you're generating well, these two should be the same. And they are very similar, okay? So it means that the things that you are generating are between themselves at the same distance than they have from the training set, which means your data are not closer to the training set than they are between themselves. So this is a good measure of generation quality. And here the last plot that you have in this notebook is the same as the one I was showing before. This is the distance to the closest natural and the energy. So you see that the more you move away from the closest natural, the more the energy goes up. This is for the generated ones. And of course, for the natural ones, the distance to the closest natural is always 0 because each sequence is a distance 0 to itself, but this gives you the range of energies that is comparable. So the generation should be done, and I can show you just the results for the PF. No, it's not done? No, it's done. Well, okay, you see this is the autocorrelation curve, this one, for the PF14, and you see that after 50 time steps, you are very close to the asymptotic limit. So in the training, we use 10, which is a bit short, but because your learning rate is small, it has enough time to decorrelate, so it's a good... and, well, once you... no, I should not do this, but... Okay, this will run again. Okay, anyways, you can run the notebook by yourself and you can check the results. So with this, you can choose your favorite protein family and you can run BMDCA. You can get the parameters, so you can do contact prediction if you don't, if you want to check the history of these things, because now you would do contact prediction with AlphaFold, but okay. But more importantly, you can generate artificial sequences and you can do what you want with them. The last thing that you can find here that I didn't have time to discuss, but can be done, is to use these models to do Monte Carlo on short times and compare with experiments in vitro. So we have a few things about it, but I didn't have time to discuss, but you find a notebook in the folder that does this, so you can run the simulation for short times and you can compare with experimental data. And well, I conclude here, but if you have any questions on these things, I'm always happy to chat, so send me an email and we can discuss.
Very good.
And Sergio said he will correct the exam at 4.
Okay. Yes, so
if you want to know.
Sergio will be here if you want to correct a discussion of his exam. You can come and discuss with him, okay? Very good. So this is the end of the lectures in the Spring College. Tomorrow we will have the exam of Francesco. And so I would like to propose one thing, because while we were here in these 4 weeks, looks like the world outside went crazy, and so maybe, I mean, so these are things there are people here coming from different parts of the world. So what happens typically is that we sort of tend to maybe discuss these things with people who think like us. So I wanted to see, maybe this is a crazy idea, but we have a session tomorrow at 5. Who wants to come just as a re-equilibration chamber, just to express different views, how we see what is going on, how we live these things, because maybe this is a opportunity we don't have normally. I mean, to have such a broad range of people. Of course, this is completely unrelated to the Spring College. This is completely informal. Has nothing to do with physics, but I think it can be useful. Okay, especially because when these things, these crazy things happen, we tend to be very much focused on the short term. But especially for you who are young, I think what is important is the long term. Okay, and then not to be too much. I mean, your trajectory not to be too much affected by. As little as possible by this thing, okay? So tomorrow at 5, for those who you want, we will be here, and it will be just, the only thing is, of course, we want to avoid any type of confrontation, so no reactive things. I mean, so we just want to, have a safe space where everybody can say what he thinks. Okay. Okay. That's it. So thank you very much. See you tomorrow at 9. Okay. Non so se vuoi